So we're going to look at experiments, the independent, dependent, key variables, and the control. So by the end of this topic, you should be able to know what is the independent variable, what is the dependent variable, what are the key variables, how do you increase the reliability of an experiment, and then what is a control. Now for IGCSE, you need to know the difference between the independent and dependent variables. So you've got other variables called key variables that you also need to know. So what are all of these? Well, if you were asked, how does light intensity affect the rate of photosynthesis? You would set up this apparatus here. So you've got pondweed, and this pondweed is going to be photosynthesizing and producing oxygen bubbles. So you need to look at the rate of photosynthesis, so you're going to look at the number of oxygen bubbles produced per minute. Now, in this experiment, you've got a lamp, which will be your light intensity, and you've got your pondweed photosynthesizing. You've also got water with sodium hydrogen carbonate, so this sodium hydrogen carbonate provides excess carbon dioxide. So what is the independent variable? Well, the independent variable is the one that you are controlling. So if you want to look at how light intensity affects the rate of photosynthesis, what will your independent variable be? It's going to be the light intensity, so you can move the lamp closer or further away from the apparatus to see how the light intensity affects the rate of photosynthesis. Then what is the dependent variable? This is going to be what you're going to look at to see how the rate of photosynthesis varies. So in this case, you're going to be counting the number of bubbles produced per minute. So if you want to be very accurate, you would look at the volume of oxygen produced. So you could use something like a graduated measuring cylinder to and look at how much is produced per minute. Now, if you were to draw your results, remember that the independent variable always goes on the x-axis. So the one that you are controlling, the light intensity, goes on the x-axis. The one that is varying is the dependent variable, and this always goes on the y-axis. So in this case, it's the rate of photosynthesis goes on the y-axis. How would you increase the accuracy of the experiment? Well, you would use a stopwatch to time the experiment. You could also use a syringe and tubing and a ruler to measure the exact volume of oxygen produced. So accuracy is about the apparatus that you are using, so you can change the apparatus to get a more accurate result. And then when you're doing an experiment, so when you're comparing one experiment to another, you need to keep key variables constant. So for example, the temperature in this experiment needs to be kept constant. How would you do this? You would use a water bath or an air conditioner to keep the surrounding temperature constant. Carbon dioxide concentration also needs to be kept constant. So in our case, we added sodium hydrogen carbonate. This made carbon dioxide in excess, so you don't really need to worry about that factor. And then the number of chloroplasts can also affect the rate of photosynthesis. So when you're using the plant material, always use the same species, the same mass, or even better, the same piece of plant. And then there's something called safety. So when you do an experiment, it's important to think about the safety. Sometimes you might be asked to describe a safe experiment. So you need to think about the risk assessment. So this experiment is a low risk experiment, but you do need to consider some safety points. For example, be careful when collecting pondweed. Be careful not to touch the bulbs when they get too hot. Sodium hydrogen carbonate is an irritant in high concentration, so use gloves when handling it, things like that. Remember that you are always asked how you can increase the reliability of an experiment. So you need to say that you need to repeat the experiment at least 3 times to calculate the average. So reliability is to do with repeating. And then finally, what is a control? Well, a control is used to make sure that no other factor is influencing your results. Only the independent variable is having the desired effect. For example, if you want to know if temperature has an effect on amylase digesting starch, you would set up 3 test tubes. So the first test tube would be your amylase and starch solution, and you would vary the temperature. Then your second test tube and your third test tube are your controls. So in this case, you've got amylase and distilled water, or distilled water and starch solution. So notice how in test tube 2, there's no starch solution, and in test tube 3, there's no amylase. Why do I have these controls? This is to make sure that any reaction that's occurring in test tube 1 is due to the amylase digesting the starch. So if you look at test tube 2, you've got amylase and distilled water. If you get a reaction occurring there, then the reaction that's occurring in test tube 1 might not be due to just amylase digesting starch solution. If you look at test tube 3, you've got distilled water and starch solution. This is to make sure that starch doesn't just randomly break down on its own. Okay, so controls are there to make a comparison so that any results that you get in test tube 1 are purely due to the reagents in there, so the amylase and the starch solution. Your independent variable is having the desired effect. And that concludes our lesson. The end.